The next experiment is study plant pigments by paper chromatography. Here aim of the experiment is to study and separate different plant pigments by paper chromatography. That is a method by which you can qualitatively show different types of pigments. So what are the requirements here? We have to take fresh bougainvillea leaves, mortar and pestle, beaker measuring cylinder, chromatography paper, pipettes, micro pipettes, very narrow tube for giving spots, wide long test tube, spillcock, funnel, petroleum ether, acetone, fine sand. These are the requirements. Then theory. What is the principle? That is, different molecules. Pigments are constituted of molecules. Different molecules, when you put in certain solution and measure its rate of movement, it moves at different rates. That it has been written, the paper chromatography is a method of separation of solutes depending on the differential solubility in two different immiscible solvents. So for that, pigments found in the leaves of the plants are taken in the paper chromatography. And what is the procedure for it? First, prathame we have to take some fresh leaves, fresh bougainvillea leaves. For example, take 2 grams, 5 grams of fresh bougainvillea leaves, cross it in 80% acetone or absolute alcohol, that is the solvent. Because chlorophyll is not soluble in water, it is soluble in these solvents like acetone and absolute alcohol. With mortar and pestle to prepare a paste. Fine sand is given here to make your crossing easier. Then it is to be centrifuged. If centrifuge is not available, you can filter it. After filtering, take a narrow strip of paper. Take a narrow strip of chromatography paper, just like this. Narrow strip of paper, we have kept it one piece there over the table and shown in the chromatography chamber also, one. So, take a narrow strip of chromatography paper, cut one end to make it a narrow wick, make a fine line. Pencil, you put a line, fine line, and then mark another line 10 cm above. Scale, we have taken the scale, after 10 cm you make another line. Say paper strip dita line you have to put. Give a fine spot, a small spot at the center of that narrow tube. से जो extract को ये dip करिवे, dip that tube in the extract and at the center of the paper गोटे मार्क देते से मार्क रे गोटे स्पॉट देबे बारंबार एमता स्पॉट देबे. सो स्पॉट देला परे with a micropipette or a fine nozzle. So then put it in the that is micropipette देई सारा परे take some time आकु टे dry हवा को देबे. Take some time. Long test tube, take a long test tube, take a long test tube like this, and now introduce the paper strip into the test tube with pointed edge dipping. Pointed edge को dip करूथिबो, just touch करूथिबो that solvent. It should just touch that solvent, dipping into the solvent mixture and keep the paper strip hanging. ताको जेमती दिया जाइछि, as shown there, you have to keep it in hanging and see how the movement is occurring, how it is absorbing the liquid. So allow the setup to stand undisturbed, just like this, that we have kept on the table for undisturbed for some time, so that it should reach 10 cm. I mean, second mark देइछे 10 cm above. सेही जगह जाए move करिया जाए देबे. So upper mark it should reach, and then dry the chromatogram. ताकू नहीं dry गोटे ये रे करिवे vacuum place रे, or in a oven you dry it. तापरे ताकू chamber रे रखी, you have to petroleum ether ये chamber रे रखी chromatogram को तार RF value movement value देखिवे. So this is the chromatographic and with these marks you will find 10 cm सबुठु बेसी मूव करथिबो कैरोटीन, जांथोपिल ता कम मूव करथिबो, ता परे क्लोरोफिल ए क्लोरोफिल बी. एई रेट रे इठी आमे गोटे रन करैथिलु. एठी प्रथम स्पॉट देइथिलु, एठी आसी 10 cm रीच करूची, इठी ये रीच करी जाउची कैरोटीन, कैरोटीन रीच करूची, ता पछकु जांथोपिल रहउची. एमती एई ऑर्डर रे रहिबो क्लोरोफिल बी, देन क्लोरोफिल ए, देन क्लोरोफिल बी. So different rate of movement you can show. So this is a chromatography experiment. आमे चारिटा पिगमेंट अच्छी, से पिगमेंट को आमे प्लांट में मेनली दीटा पिगमेंट रू क्लोरोफिल ए बी अलग हो जाउछन, जांथोफिल कैरोटीन भी अलग हो जाउछन. सो ताकू डिफरेंशियल तांकर मूवमेंट जेहेतु ये डिफरेंट मॉलिक्यूल कॉन्स्टिट्यूट करूछन, सिंस दे आर मेकिंग कॉन्स्टिट्यूटिंग डिफरेंट मॉलिक्यूल्स, तांकर रेट ऑफ मूवमेंट डिफरेंट. क्लोरोफिल बी रो सबुठु कम मूवमेंट, तापर रहइबो क्लोरोफिल ए, तापर जांथोफिल, तापर कैरोटीन. इन दिस वे you will find, and coloration you will find like this: yellow color, orange, yellow orange, that is carotene, yellow xanthophyll, dark green chlorophyll A, and yellow green chlorophyll B. Different plant pigments separate into different colored bands. So different band upon paive. The orange yellow band corresponds to carotene, yellow to xanthophyll, dark green to chlorophyll A, yellowish green to chlorophyll B. से सेठी सेमती आमर हेइतला ग्लाजुअर से कलर मैनेज करी गलानी, माने उड़ी गलानी. ये नले you have to write the precautions, and you have to write in this way. This is the chromatography experiment.